Hey guys, welcome to our exam 1 review for lab. So in this video I'm going to be pointing out some key points. Also just remember that this review video is not an exclusive list of everything. This is more or less just to get your thoughts in order. So I'm going to be walking through our study guide in this video. So let's go ahead and get started. Actually, before we get started, I want to point out some stuff that I added onto Canvas. So if you go to our homepage and you go to modules, what you need to do is find our module for our exam 1 for lab. So exam 1 lab. So if you go under here, I did add quite a bit of stuff. So I added the guide as well as the 3 PowerPoints for our labs, and then also some videos for y'all to watch. So here's a lab safety video, experimental design and everything, and then also how to make wet mount slides. So definitely use that to aid in your studying. So let's go ahead and get started with our guide. So our first question is, what are the 5 main lab safety rules? So I know I didn't go too much into lab safety with y'all, but these are pretty basic ones. So the first one is, so I'm gonna go ahead and type in red for y'all. So it's always proper lab attire. So lab dress. So always look the part. Also make sure you wear your personal protective gear. So like your lab coat, goggles, stuff like that. This one we did not talk about, but you always add acid or you add base to your solvent, so to your solution. And then also know where and how to use safety equipment, such as like the lab shower, the eye wash station, know where the fire extinguisher is, and so on. And then also proper lab behavior. So no horseplaying. So like no running around. Also, you can see all of these lab safety rules inside that video that I posted onto Canvas. So the zombie college. It's pretty cool. It's pretty simple. It's pretty entertaining. So go ahead and check that out. Okay, so now we're going to be doing some conversions. So on the test, you need to know how to actually do conversions. There are some conversion factors that I will give you, such as the kilogram to pound, and then also how to calculate Celsius to Fahrenheit. I'll give you that equation. But whenever it comes to like the metric unit, you need to know how to convert in between different units. So we're going to go ahead and work through some of these. So what I'm actually going to do, I'm going to copy and paste these into a different document so it's easier for me to write on. So 1 second. So this is going to be conversions. Okay. So let me just enter through all this. Okay. So I'm going to make this a little bit bigger for y'all. Okay. So you always start with what you have. So in this case, we have 34.7 meters, and then we want to get to centimeters, so we're going to go to centimeters first. So what we need to do is put our unit of desire on top, and then our unit that we want to get rid of on bottom. So now what you need to know is, what is the conversion factor between them? How many centimeters are in every meter? So for every 100 centimeters, there's 1 meter. And then from there, you'll just multiply across. So 34.7 times 100. I don't know that. Let me pull up a calculator, guys. Okay. So 34.7 times 100. So 3470 will be our answer. So it's 3470 centimeters. Do not forget your units, guys. I cannot stress that enough. Please don't forget your units. Okay, and then you'll do that for every single one. So I'm gonna do one for each of these, okay? Okay, so now we have 9.2 centiliters, and we're gonna convert it to liters. So we need to put centiliters on bottom and then liters on top. So for every 1 liter, there is 100 centiliters. So in this case, what you'll do is that you'll still multiply across, but then you also need to divide by the bottom. So it's gonna be 9.2 over 100, and then that will give you your answer. So 9.2 divided by 100, so 0.092, so 0.092 liters. Okay. Okay, so our next one, we have 1562.3 kilograms. And let's go to, I'll go ahead and do the gram ones for y'all. So we need to get rid of kilograms and we want to go to grams. So how many grams are in every kilogram? So for every 1 kilogram, there's 1,000 grams. So it's going to be that number times 1,000. So with your numbers, guys, if they do ever get really big, whereas like this one is pretty big, it's in the millions. You can definitely put it into scientific notation. That's perfectly fine with me. But if you do scientific notation, it needs to be proper scientific notation. So for this one, the proper scientific notation will be as follows. Okay, so make sure you're doing proper scientific notation. Okay, so I'm gonna leave the rest of them to you guys, but definitely know your metric conversions. So that's just an example of some of them. I'm not gonna do all of them. Okay, and then the next question it asks for is, what are the appropriate tools whenever measuring the following? So if you're doing length, you need to make sure that you're using like a ruler or like a meter stick. And then if you're doing volume, the best one is graduated cylinder. I don't even know how to spell graduated. Graduated cylinder. That is the best one. That's the most accurate of all the glassware to be measuring in. Or if you are taking small amounts, so for small amounts, you can use a pipette. That's also acceptable. For mass, you're using a balance. And for us, we use a digital balance, but if you just put balance, that's good. And then temperature, we are using thermometers. Okay, so it's pretty self-explanatory stuff, at least on number 3. Now with whenever there's differences between independent and dependent variables, and then also like positive, negative controls, difference between experimental and control groups. This video right here, the first one that says scientific experiments, this goes over all of it. So I'm briefly just going to, I'm pretty much going to copy and paste something that I've already written real quick for y'all. Just so y'all can see it. So now the difference between independent and dependent variables is as follows. So the independent variable is the one that represents the one that's actually being manipulated. So what are you changing in your scenario? Whereas your dependent variable will depend on the outcome from the independent variable. So definitely be able to distinguish between the two. And then with positive and negative controls, a negative control is a control group where it's going to give you a negative result. So it's results that you are expecting to not produce anything. Whereas a positive control is one where you know that it's going to produce results that you want. And then also the difference between control and experimental groups. So a control group is for comparison, and your experimental groups is everything except for your controls. So this is where something will differ from the control group. So like we did the Benedict's test last week, I believe. So your control group was like a negative control group was your water. And then what you did in order to change it to make a group experimental is that instead of adding water, you added like sugar water or something like that. But definitely look over in that video. It's pretty quick. It's really quick and dirty, that video. It's pretty self-explanatory. It's very simple. She definitely simplifies it very much. Okay, now with graphing data, so I am going to give you some data and I am going to want you to graph it. So from this, you need to be able to recognize what is your independent and what is your dependent variables. So I'm going to copy and paste another graph that I had. So with your data, it should look something like this. So you should have axis titles. So in this case, time is going to go on the x-axis and then mass is going to go on the y-axis. And then you need to also have a title for the actual whole graph. And then what you're going to do is that you're going to distinguish between kitten 1 and kitten 2. So it should look something roughly like this. On the test, if you don't have color, just like say like circle is kitten 1 and square is kitten 2. But definitely if you want color, I can definitely provide another color pen or something for y'all. Now it asks the question, which one grew faster of these kittens? So from that, you need to do some basic math. So in order to calculate who is growing faster, you need to calculate slopes. So that's going to be the change of y over the change of x. So that breaks down to y2 minus y1 over x2 minus x1. So then with that, you're going to do like kitten number 1 over here. So, and it wants between week 3 and 4. So you're going to take the data from week 3 and week 4. So it's going to be as follows for kitten number 1. Okay, so that was the change in their weight over a week time frame for kitten 1. Now for kitten 2, it's going to be as follows. So according to this, the kitten that grew faster was kitten number 1. So kitten number 1 grew faster in the time frame of week 3 to week 4. So on the exam, I will expect you to be able to do this, something very similar to that. It won't be the same data, but it'll be the same principle. Okay, so next it asks like what are the different reagents and the type of molecules that it tests for, and be able to recognize a positive and a negative test. So that will bring us to the PowerPoint where we identify macromolecules. This one is a fabulous PowerPoint. So like on the exam, what will happen is that I will give you something like a picture like this on the exam, and then I may say like, okay, so it was testing for simple sugars or simple carbohydrates, and I would want you to be able to say like test tube A is the negative control. So like if I put like this is test tube A and then this one is test tube B, I would want you to be able to tell me that Benedict's was the reagent that was used to test for simple carbohydrates, and then also tell me test tube A is the negative control, test tube B is positive. So that's just for Benedict's, but that would be for all 4 of them that we ran. So we did Benedict's, we did iodine, we did burette, and then we did Sudan 4. So I would definitely want you to go through this PowerPoint and look at the positive and negatives throughout this test. So the likelihood is that I'm going to use pictures very, very similar to this, so it's very easy to tell. So I'm not going to give you one that's like, don't know, okay? So definitely be able to identify that. And this PowerPoint is really good for question 8. Okay, so now it says to label the parts of a microscope. So let's make this bigger. So let's go ahead and walk through labeling this microscope. So A is the eyepiece. B is the rotating nose piece. So this is the one where it actually moves the objectives. Sorry guys, my handwriting's terrible on this. And then C is the actual objective lens. So I'm gonna abbreviate. D is the stage. So this is where you put your microscope, where you actually put your microscope slide. And then E is the condenser. So this is the one where the light will shine through. So how much, do you want to concentrate your light or not? And then where are we at? We're on F. So F is the base. Oops. So base, this is just literally how it stands up. G is the arm of the microscope. H is the coarse knob, so this is the one that brings the specimen initially into focus. And then I is the fine focusing knob. J is pointing to this region right here. So J is the stage controls. So these stage controls are what actually moves the microscope slide on the stage. And then K, oops, oh no, why isn't it writing? There we go. So K is how bright do you want your light? So that is the light intensity knob. Okay, so definitely be able to label the parts of the microscopes as well as what is their function. So definitely be able to recognize that. Okay, so now on number 10 is how do you make a wet mount slide? So there are roughly 3 main steps on how to make a wet mount slide. And that involves you get your slide. What you do is that you add water. Oh, let me write in red for y'all so you can see it a little bit better. So what you do is that you add water to your slide, and then what you do is that you add your specimen. So add your potato or onion, whatever it may be, to the slide, and then you place a cover slip over it. Okay, so you'll get practice doing this in lab, so definitely remember how to do it. And then also, whenever you are preparing your slide and once you observe it, what you need to be able to recognize are different cell types. So if I were to give you a picture, a picture like this, what I want you to be able to tell me is that it's an onion cell, and then also be able to label the parts of the onion cell. So this is the cell wall right here, this is the nucleus, so be able to identify that, and then also the clear area in between is the cytoplasm. And then be able to tell me what stain did we use in lab, and in our lab we're going to be using iodine, so Ludol's iodine. So I want you to be able to do that for all of them. So that's the onion, be able to recognize the potato, the elodia, be able to recognize the elodia, and then also human cheek cells, and then some bacteria cells like this, so the different types of bacteria cells. So be able to recognize those on the exam, and then be able to tell me what stain did we use on the slides that we actually prepared. And then be able to label the parts of the cell. So like on the potato, be able to label these amyloplasts. So this is where like all the starch is stored in a potato. So be able to identify that the purple region or like the brown black region is the amyloplast, whereas the white area is the cytoplasm, and then the border between them is the cell wall, and so on. So be able to do that for all cell types. Okay. So that completes number 10. So now we're going to talk about number 11. So number 11, know the concepts. So ideally, theoretically speaking, that if you have higher temperature, then you have increased energy. So let's write that out. So for this, if you have higher temperature, ideally that means that you have higher energy. So meaning that you have more movement, which leads to more movement of molecules. So if you were to have just a beaker of water, put a drop of food coloring in it, so if you had a hot beaker of water and then a room temperature beaker of water, theoretically speaking, the hot beaker of water should be able to become a different color quicker as compared to the colder one. And then the molecule that's actually allowing this to happen, for diffusion at least, is water. So water is the main molecule that we are going to be talking about. Okay, so that's pretty much all I want you to know for like osmosis and diffusion. And then one last thing before we wrap this up is the eggplant. So we touched base about this in class, that osmosis worksheet that we did. So definitely refer to that. It's a very good reference guide. But generally speaking, whenever you have any plant cell really, but eggplant is just easy because it's nice and big. But if you have an eggplant and then you're going to put it into unknown solutions, so generally speaking, if the cell, or like the eggplant, so I'm actually going to type eggplant. So if the eggplant increases in mass, then you had a hypotonic solution because the eggplant got bigger, so it swelled up. Whereas if the eggplant decreased in mass, then you had a hypertonic solution. But then if the eggplant's mass remained relatively the same, then you had a isotonic solution. So how would you be able to determine if you had that? What you would do is that you would take the initial reading of the mass of the eggplant, and then after it's been in the solution for so long, then you would weigh the eggplant again to see, and then depending on how the mass differed from initial to final is how you would determine what solution type did you have. Okay, so that was a pretty quick general overview of our review. Like I said, it is not comprehensive. Definitely, if you have any questions, let me know. But that is just a quick nitty-gritty breakdown of the review, and then also some basic techniques that I will want you to know. So like on the exam day, it will be a two-part exam. There'll be a hands-on portion, and then there'll be a written portion. So whenever we have our lab exam, definitely come prepared, be in lab attire and everything, be ready to go. So what will happen is that we will have stations, and at the stations, I will have different setups, and then from that, I would want you to answer the questions accordingly. So like at the station, what will probably happen is that I'll have a microscope set up. I would want you to be able to identify your cell type, be able to like draw your cell type or something of that nature, and then also be able to measure out a certain amount of volume. Something like that is what I would want for like skills wise, but I'm not too sure yet, guys. But it will be something of that nature. It won't be anything too crazy, I promise, so don't panic. And like I said, if you have any other questions, just let me know. If not, thanks for watching, guys.